We're going to continue with the focus on emerging therapies theme, and we're very fortunate to have Patty Seymour from Bioprocess Technology Consultants, who's put together an expert panel here, folks, to really take a look at the emerging biotherapies and their manufacturing challenges. As publishers in this industry, we see the trends that are happening sometimes more in the aggregate than a lot of folks. And what we do see is that while the science of these emerging therapies are progressing rapidly, the technologies are starting to lag behind a little bit. So when we get to scale and we get to volume levels, we're not quite there yet as far as allowing these therapies and these emerging therapies to get to commercial scale. So the panel that Patty's put together here includes Mark Angelino, who's the Senior Vice President of Pharmaceutical Sciences at Bluebird Biotechnology. And if you were here earlier in the theater, Andreas Wheeler, PhD with Lonza, is the Global Business Unit Head of Emerging Technologies. And joining us also is Dr. Chris Chen from WuXi Biologics, who's the Chief Executive Officer. So without further ado, I'd like to introduce Patty Seymour. The title of the roundtable is The Emerging Biotherapies and Their Manufacturing Challenges.
All right, well, thank you. We're really excited about this topic. I think there's a tremendous amount of interest right now in the amount of different types of emerging therapies right now. I think we're really starting to get beyond the monoclonal antibody issues that we've been working so hard on solving for the last 10 years. And I think now we're kind of at the approaching the next frontier in terms of these new emerging therapies. So I'm really pleased. I think we have an all-star cast here of really deep expertise who can talk about a lot of these topics. And so we'll really look at several areas. So again, we have Bluebird, who is really preeminent in the gene therapy area. Lonza also, which has a wealth of capabilities across emerging technologies, including antibody, cell therapy, gene therapy, and WuXi Biologics as well, similar, wealth and depth of expertise around multiple different platforms for manufacturing. So again, thank you, gentlemen, for agreeing to participate on this panel. Just have a few slides, very briefly, to set the stage of what we're talking about here for this particular session. So in cell therapy, there are two basic types of cell therapy that we're looking at: the allogeneic or the autologous. Allogeneic, coming from the patient; the autologous, somewhat immortalized types of cell lines. Each of those have different types of concerns regarding manufacturability. So hopefully we'll be able to touch on some of those issues. Gene therapy. So next, the different types of platforms as well. The viral vector therapies, again, have been around for quite a while. There are different ways of approaching that. And then I think some of the really exciting technologies that we're really just starting to deal with from a manufacturing perspective are the gene editing technologies. And so hopefully we'll be able to dive into that a little bit more detail. And then lastly, the antibody drug conjugates. Again, those have been around for a long time, still sort of waiting to maybe fully emerge from the development, but there's certainly very interesting technologies surrounding the conjugates field. So next, just to give a little bit of context here around the different types of cell therapies that we're talking about. Often the manufacturing aspects involve harvesting cells from a patient. And so what that entails are these little mini manufacturing environments that possibly need to be built up around processing these cells and doing the transformations or whatever that needs to be done to the cells to reapply the therapies back to them. So there's a host of issues that we need to be aware of, the supply chain issues, the control issues, the quality issues, et cetera. And we'll talk, hopefully, in some detail around the concerns we have with these types of technologies. So, again, we have some real experts here in working on the cell therapy aspects. But, to date, these technologies are, unfortunately, fairly open. They're very manual, relatively small scale, done at a research grade. But we really do need to move it towards a more robust process, a larger scale, something that's reproducible, and of course it all does have to be done according to GMP. So then moving on to gene therapy, again, very, very, very exciting field right now, and looking at the two classes of gene therapy, whether using an AAV or a lentivirus platform to deliver your therapies. This slide was taken from a BPI article that was published very recently in April of this year. So if you haven't had the opportunity to read it, just go onto their website and download it. It's a really wonderful overview of the gene therapy space. But it does show here the number of drugs and their indications that they're going after. So you can see it's a very active field, looking at multiple types of indications, and you know, anywhere from a handful of drugs to up to 10 competition in going after each of these indications. So certainly an attractive field for a lot of product development companies. Okay. And again, you know, some of the manufacturing issues surrounding gene therapy here are, it's not quite as simple as the antibody manufacturing. You need to create the vector, amplify it, expand it, purify it, do some polishing, clean up at the end, and then do the fill finish. And so I think a lot of the focus maybe today might be around the drug substance part, but the drug product manufacturing has its own issues because many fill finish houses aren't willing to accept this type of technology or this type of product into the facility for cleanability, et cetera. So that becomes an issue then as to how to find a manufacturer for that part of your supply chain. Again, maybe not technically difficult, but becomes an access issue. Okay, and last, drug conjugates. Again, we've been talking about this for a long time. I started my career at Immunogen back in the 80s, so that says, you know, how long this has been going on for. So I remember when the maytansinoid technologies first came out of it, and you know, working on it now. You know, it's a commercial product. It was very exciting then. It's still very exciting. But you know, when is the next product going to be approved using whether it's the Immunogen platform, the cytogenetics, or a host of many other types of toxins? But part of the challenge is these types of products are incredibly complex supply chains, have multiple parts to it, and it makes them very expensive to manufacture because of all of these different parts. An ADC essentially is the equivalent of 4 to 5 CMC programs. So if you know how much it costs to develop an antibody in a CMC platform, then you add the toxin to it, and then some of these other components, you can see the cost of goods starts to increase substantially. And so that often becomes an issue in a portfolio management scenario when you're rationalizing, can you make a drug cheaper with using a different technology to achieve the same therapeutic end? Okay, so that's it for the slides. It really just was intended to give an overview of some of the technologies we want to talk to. It doesn't mean we're limiting it to that, so certainly when we go into the Q&A session, if people have other technologies they want to talk about, then please feel free to ask. So now I'd like to turn it over to the panel for a few minutes. I would like them each just to give a couple of minutes description around, you know, the companies, you know, what kind of technology you're currently working on, and then we can go into some of the issues that we're seeing around the emerging therapies. So maybe, Chris, we could start and just go down.
Yeah. So, my name is Chris Chen. I run the biologics division of WuXi AppTec, and as Patty mentioned, that WuXi provide integrated service for ADCs, bispecific antibodies, traditional antibodies, biologics, cell therapy, and gene therapy. So we see a lot of challenges, as Patty just outlined already. I think it would be an interesting panel to learn from the other panel members and from the audience. Thank you.
Thanks, Chris. That was a very fast introduction. My name is Andres Waller. I work for Lonza. Who in the room doesn't know what Lonza is? Who Lonza is? So I assume you don't know? Okay. So please listen. Lonza was founded... Okay, perfect. So Lonza was founded 120 years ago in the middle of the Swiss Alps, a beautiful area. You see the Matterhorn in the south, so worth a trip visiting Lonza. And Lonza was one of the first biotechnology provider. They moved into the biotechnology business 30 years ago. 10 years ago, they moved into cell therapy. Plus, they built a bioconjugate facility in Visp in Switzerland. It was a huge investment at that time. Think about it. At that time, there were only 30 ADCs in the clinical program. So quite a remarkable, courageous decision to build such an expensive facility. But it brings the leverage of the biological experience and the small molecule experience Lonza has perfectly together. As we saw in antibody-drug conjugates, you have a biological component and you have a small molecule component which you couple. And that's the beauty of these molecules. Plus, 6 years ago, Lonza acquired Vivante in Texas to move into the gene and viral therapy space. And since then, we've manufactured multiple batches. We've commercialized a few products, but maybe more about this later. Thanks a lot for being here.
So the company I'm working for is Bluebird Bio. So Bluebird is, again, a younger company, so 6 years old roughly. It is a gene therapy, so we qualify as ex vivo lentiviral vector autologous gene therapy. So what that mouthful means really is we work with lentiviral vector to transduce what are patient-specific cells. We provide them back to the patient, and the assumption or the hope is we're providing a curative therapy. So we have 4 products in the clinic, one around a CNS disorder for young boys, one around thalassemia, sickle cell disease, and then we are in oncology, so chimeric antigen receptor space. In multiple myeloma, we have a study. And then to boot, if the complexity of the manufacturing platform wasn't difficult enough, we've bolted on a gene editing platform a couple of years ago, a company we bought. So we also have a gene editing play, which again is more portfolio coming along preclinical.
Great, well thanks. So that gives you a sense of the different types of companies, of product focus, CMO focus. But one of the things that really want to open up, and maybe Mark, we can start with you on this question, is what are the patient demands and needs for these new therapies? So obviously Bluebird saw an unmet market potential and created a company around this. So maybe explain a little bit what you're seeing in your patient populations and what you're doing to try and meet those demands.
Sure. So again, it varies depending on the indication, right? So when we started 6 years ago, early indications were ultra-rare diseases, right? So stem cell-oriented, ultra-rare, where, again, our lead in CNS, you're talking, worldwide prevalence is about 50 to 100 patients a year, all right? Now, if you go to thalassemia, which is next, you're probably an order of magnitude more. When you go to sickle cell, which again is a little further back in our pipeline, probably another order of magnitude more. And then, essentially in oncology, you may get into the thousands, if not, potentially tens of thousands. So, our approach has been almost a stepwise gradation, right? So prove the technology with what are smaller indications. We do feel that there's a market for those indications. Again, I think the reality is it's probably several of those indications as we start thinking about growing the company, thinking of infrastructure. But there are bigger indications to be had, and we're seeing that as well. And again, I think the industry is seeing that as well in the last few years.
Okay. Andreas or Chris, any thoughts on where you're seeing from your customer base, what the patient demands are and what they're coming to you to try and fulfill those demands?
When we look into these patient demands, when you look into chemotherapy right now, it's obvious that when you treat a patient, first-line treatment, chemotherapy, you fight not only the disease, you fight his whole body. And when you look into some of the targeted new approaches, it's really obvious that when you treat a patient with an ADC, for example, the patient has a lot better life. He has a lot less side effects because you're just targeting the antigens on the cancer cell which you want to destroy. And this is significant difference to previous treatments where you really fight the body. There's another nice approach which we see more and more with these treatments is that you can actually in principle enable your own immune system to recognize the cancer and destroy the cancer. And this is the beauty of these drugs, so you have a lot less side effects. You encourage your own body to fight the cancer. This is a totally different concept, and in my opinion, it's a total paradigm shift. On the other side, what we see with a lot of these drugs is that there's, of course, a lot smaller patient population treated, as we talked about, rare diseases, ultra-rare diseases. So the interest from big pharma is significantly lower. But on the other side, it's the beauty of some of these treatments is that they might be in the future or maybe already cured. This is a completely new paradigm as well.
Yeah, I guess for cell therapy and gene therapy, the patient population may be relatively small. But for complex biologics, the ADCs, the bispecifics, actually the volume may be as large as antibody or maybe even more, right? Theoretically, hypothetically speaking, for ADCs, the dose may be lower. But in the clinic, we haven't really seen that. We've actually seen sometimes to get the maximum effect, people are actually even using higher dose than traditional antibody. So if some of the ADCs are becoming really successful and the volume can still be as huge as you're talking about maybe 500 kilogram or even a metric ton scale of ADC, that will be a huge burden on supply chain. So I think right now, I think globally the capacity for ADCs are not even there. So it's actually going to be an interesting proposition in the next 5-10 years to see if ADCs to continue to become successful and become blockbuster product with multiple of them, and then there will be a huge shortage of the ADC capacities, commercial manufacturing ADC capacity, both the drug substance and the drug product side. And for bispecific antibodies, it's very similar. So it's a bimodal distribution. So for cell therapy, gene therapy is relatively patient-derived and relatively lower volume. But for a successful ADC and bispecific, it could be another blockbuster. You're talking about hundreds of kilogram or even metric ton scale.
Yeah, no, it's great. And you know, the point is that a lot of these technologies are really getting past sort of the pellet gun approach of chemotherapy, where it just kills much of any rapidly dividing cells available and just kill the cells that you need to kill and have it very focused. And so I think that's what's one of the very attractive things, make it so specific so the patient has the best benefit with the least side effects as well. A lot of that, though, is still a work in progress, certainly with the ADCs, some of the drug antibody ratio and the free toxin issues can become problematic, and then it's certainly with gene therapy, how focused is it? Is it only going into the cells that you want versus to other cells? So I think that really helps clarify, what are the limitations of some of the existing medicines, and that really is driving the development of some of these new therapies. So I think, the next question, and again, looking into your crystal ball, but also being pragmatic. And, with gene therapy, Mark, you mentioned that a lot of your products are truly intended to be curative, but are they in fact? I mean, will they be curative? And what does that mean? And I think of it from a manufacturing perspective, a product perspective, a reimbursement perspective. These are big issues because if you cure your patient population, then you don't have a product anymore. And then maybe talk a little bit about that and how, are there some of these where you might actually have to reapply because for whatever reason it doesn't, the cells are no longer being effective or expressing what you want them to express?
Yeah, it's a good question. I mean, so in general, the field, right? I mean, the field we've seen over the last 10 years, some cases where, it's been a persistence issue, right? So again, I think the hope is getting there, right? The hope is getting to where it may not need the reapplication. I think when it comes to the forecasting, right? So this question of capacity, right? It depends, right? So there are cases in our product portfolio where you still have a prevalence associated with this disease, and so in essence, what you will have is a peak, and then you'll have a plateau, right? To what is essentially treatable population. In other cases, what you're seeing is very much more traditional pharma approach to the forecasting, where your peak is much higher and then you're tailing down, and a lot of that is because some of these are not acute. So, for example, one of our indications, these young boys may die within 2 years. So there, your typical forecast will peter out to what is the prevalence. In some cases, what you have is that it is a disease of a lifetime where essentially morbidity may set in around 30, 35, and so your forecast is much more typical, where there would be a peak 4 or 5 years out, because you're treating what is pent-up demand within the system, and then eventually you peter down to what is prevalent. So I think a lot of that has to deal with how you set up capacity. It is not a single drug to fill a capacity in a pipeline, right? It is a much more traditional model of how does peak and trough really overlay and really start letting you use capacity wisely. So again, that's a lot of stuff that we deal with on a day-to-day basis, or on a week-by-week, month-by-month basis, just as we feel what we need from a portfolio to make it sustainable. So, but again, I think that's really the question to see. I think, and the data sets are always so.
Yeah. Okay. And then I guess from the CMO perspective too, listening to sort of the product company approach about how you're forecasting, how would a CMO then build infrastructure or a support service platform around that, given the uncertainty in demands and potential peaks and fluctuations and stabilization over time.
Yeah. Maybe very quick, when we look into this industry of cell therapy and viral therapy, plus the bioconjugates, we see around 1,100 products are in the clinical pipeline right now, from preclinical up to commercial. I don't know if people know here, but how many commercialized products are on the market in cell, viral, and in bioconjugates? It's actually less than a dozen. And then plus what I said earlier, 95 of these drugs are innovated by smaller players like a Bluebird. Bluebird is not a small player anymore, but there are some even smaller players. And so I think this has huge implications for this market, and as a CMO, you have to look into the pipeline. You have to look into what's in the preclinical pipeline right now to prepare yourself and build the right capacity to serve these clients. And I think if you do this courageous model, if you invest right into cell and gene therapy, plus into bioconjugates, you can lose, but you can also win big. When Lonza invested in 2006 into bioconjugates, nobody else invested into bioconjugates. And they invested 50 million, but at the end of the day, it played out. But it could go all the other way around. So I think as a CMO, you have to take a certain risk, and I also think you have to really look deeply into process innovation. That's your strength. And there's maybe one of the paradigm shifts as well coming up. When you look into these new products, the IP is not only in the product, the IP is in the process. And this is very, very different to many of the other drugs on the market. So as a CMO like WuXi and Lonza, you have a big advantage now because the process innovation is absolutely critical to the success of the innovators in the market.
So Andres put it very well. I think it's as a CMO, I think we manage capacity, but it's actually much less risk compared to a product company, because product company, even if you have a dozen product, the product could potentially fails. But we manage the entire portfolio, and as Andrea said, there's a lot of program in there. So I think WuXi's approach is we are very confident. Our approach is build, they will come. So we're investing in antibody manufacturing capacity. We're investing in ADC manufacturing capacities. I think as long as we're bullish on the industry, I think the payback will be here. I think it's really a matter of eventually you have a leap of faith to go through. So for ADCs, we still believe in ADCs. And we actually, as Patty mentioned earlier, the ADC supply chain right now is a nightmare. You may have 4 or 5 CMC programs. WuXi put everything within 100 miles. So we have a warhead synthesis facility, which is globally certified. We have an antibody manufacturing facility. We have a conjugation facility. We have a drug fill. Everything within 100 miles. Essentially, WuXi can manage the entire ADC supply chain in a very short distance. I think that's to the extent how we integrate everything, testing. Essentially, from a concept to the IND or eventually a BLA for IND, everything happening in a very limited distance.
Yeah, and that's actually good, Andres. I want to touch on a point that you made around the process is defining the product to some degree. Are we actually going backwards? We've made so many strides on well-characterized biologics for proteins, where the process doesn't define the product per se anymore. But now, on some of these therapies, are we actually going back to the old model where the process actually does define the product? What are your thoughts around that? Maybe you can elaborate a little bit more.
I'm not a regulatory person to answer that question, but I clearly think when you look into the autologous space where you have patient specific treatments, you can think in principle of 2 business models. You have a centralized business model where you take the blood of a patient, you change it, you genetically modify it, you grow the cells, and you transport it back to the patient and re-inject it to the patient. So with the advantage that you have everything centralized, meaning the QC, QA release. But on the other side, you could think of a decentralized model. If you have a fully automated reactor which can do the autologous cell manufacturing, you could have these centers at the hospitals itself. And then you could treat the patient at the hospital, take the blood, change it, and re-inject it by this fully automated processing. And there is definitely, we are not there right now, but we are moving fast into this direction. to offer clients both business models, centralized and decentralized model.
Yeah, and I think actually that is probably one of the areas I think manufacturing professionals really need to focus on, because when we talk about these big facility expansions, hundreds of thousands of square feet, tens of thousands of liters for proteins, but now we're going back into, we need a hood. We need a few hands and benches to actually create these little manufacturing environments in these local environments. It's really, truly a personalized medicine approach. And all of the regulatory controls around that, I think, are still being sorted out. Globally, from the U.S., Europe, and within these facilities and the companies, how do you deal with that? How do you define a batch? How do you release a batch, et cetera? So maybe Mark, are you dealing with some of those things and sort of those micro manufacturing environments? And what are some of the really challenges that you haven't been able to crack yet?
Yeah, I think it's all about challenges right now, right? It's early days, right? So, getting back to an earlier point around product versus process, right? It's the bane of my existence, right? We've spent so long trying to get away from that, and I think right now it's early days where I think there's conservatism which says, okay, well, we don't change the process and we won't affect anything. I think that's dangerous, right? We've had discussions with the agencies where, you can't do that. The agency does want you to, the agencies do want you to evolve the process. It has to evolve, right? The technology is changing so quickly, and so how do you really start defining and working with the agencies around, okay, it may have changed, but fundamentally what has changed? And so, again, we're getting more latitude as the science catches up. Again, I think the challenge, is going to be dependent on the indication, to be open. We've had some ultra-rare indications, we've got some broader indications. The ultra-rare indications will not afford that sort of complexity, right? Typically in these cases, those children, those people afflicted are going to the world's experts that are probably 5 or 6 people in the world. And so in essence, you have to be there. Right? You don't have to be everywhere. I think as you start getting into these larger indications, sickle cell, as you start getting into oncology, the expectation is to have a much broader footprint. And there, again, it's the balance. We face the balance. We don't know the answer yet, but it's this question of, smaller footprint gives you more control versus a broader footprint versus essentially parabolic complexity that's associated with really trying to get a system like that working. To your point, Andreas, you have it at every bench, but are you going to be able to control it at every bench in the same way, which is always a concern, right? So again, I think it will depend on the indications and depends where we get with the technologies. Again, that is still evolving over the last year or two.
So that makes me think of process control. For example, do these types of technologies and manufacturing platforms lend themselves to really starting QBD very early, from the very, very beginning, as opposed to, more organically until you have a proof of concept, and then sort of tightening the nuts and bolts around it, sort of in a latter phase of development? Where do you see that? If you can define all of that early and then go out into these mini manufacturing environments, do you think that gives more flexibility? Or even if you have to set up regional manufacturing facilities for any number of reasons? Mark, and then we come down to Mark, Andreas, and Chris.
Yeah, again, I think part of this is, I think we've all grown up with QBD now. In a more traditional setting where, again, I come from a more traditional setting, there's an expectation to use it. I think where you're dealing with some of these therapies right now is, think of a stem cell therapy. What specifically is the long-term populating stem cell that you want to modify, right? So I think there's an element where you can do QBD, and I think there's a lot of investment in trying to learn as much about your product. There's an element where the science hasn't quite caught up to exactly what is that piece that you're trying to control. So I think, at this point, I think a lot of the folks in the field are learning as much, trying to acquire as much information. I think there's still a little bit to learn, quite a bit to learn about actual impact on the product attributes at the end game. So again, I think it's a bit of both worlds right now.
It's a good point. QBD, right? Can you hear me? Okay, perfect. When you look into how cell therapy is done, it's done on a two-dimensional field, and you need huge facilities and a lot of manual processing. And to move these processes into a 3D process, meaning into a bioreactor, it needs a lot of QBD, but also at the end of the day, you need to show that there's comparability, that the cells you have grown on a 2D platform have the same, let me say, appearance than the cells you're growing in a 3D bioreactor. And so we are right now doing a lot of QBD programs by transferring a lot of our 2D processes into a 3D process. And to do that, you need, of course, special microcarriers. You need to look into a lot of process innovation around this. But at the end of the day, you have to show that the cells are the same cells than you have grown on a 2D platform. And this is a challenge right now, but we're making big progress.
I think if you combine the sort of the risk-based and QBD, it's probably make a lot more sense. For smaller indication, ultra-rare disease, you probably don't have all the resources to apply QBD in the beginning. And for the much larger indication, probably QBD probably make more sense.
Yeah, and I think that's a big part of the service offering. Can CMOs actually offer the QBD process? It can be very labor intensive and costly, unfortunately. But what I'd like to hear, what are some of the other offerings that CMOs are building into their business models to meet some of these demands? I think we touched on a little bit with the ADCs, but Andres, maybe the 2Ds versus the 3Ds. Is Walkersville going to start having 1,000-liter bioreactors to do cell therapy, et cetera? And where do you see that? How is that going to change? And how do you look at your manufacturing networks to do those types of things?
First of all, we don't need a 2,000 or 1,000-liter bioreactor. I mean, just to give you a dimension, in a 200-liter bioreactor right now, we can manufacture as many stem cells as we can do in a huge facility, a football field, huge facility, if we do this in a 2D process. So this is a huge improvement, and that's the future for many of the allogeneic processes. On the other side, we see, especially with CAR-T, that there's a lot of autologous processes entering the market, and we need an innovative solution how to manufacture these processes. Because each patient has his own process in his own reactor, and so you need to think about how can we guarantee consistency of the batches, how do we talked about it, manage the supply chain. So there's a lot of things we are thinking about, and as I said earlier on, it is a strength of a good CMO to look early enough into process innovation. I don't think a CMO should compete with the innovators, but a CMO should enable the innovators to bring the drug to market as fast as possible. And this is another very important point. I don't know, will you talk about this later on, about the fast track?
Yes.
Okay, excellent. So I'll shut up now and leave it to Chris.
As Andreas made it very well, I think CMOs or CDMOs like WuXi now began to offer solutions. It's not only just help you with one piece. For some of the smaller companies, we actually provide guidance from beginning of the program all the way to essentially do the process design for you, do the process implementation, manufacturing, eventually scale up plan. Essentially help some of the smaller companies, guide them through the entire product, almost like product life cycle approach. So that's for the cell therapy and gene therapy. For ADCs, we actually even offer the discovery service. So we can help you assemble different type of warhead, different type of conjugation chemistry, or different type of antibodies to essentially make you 40, 50 different ADCs. And you can use that ADCs to do pharmacology studies to pick out which are the best one. And then so we essentially start from 1 milligram to 1 gram to 100 gram, eventually a kilogram.
Maybe, Patty, very quickly, maybe a little comment on the ADCs, because we touch it all the time. I don't know if everybody in the room is familiar with ADCs, how they look like, and what's the dilemma of the ADCs is. Because if you think about, you have this huge antibody. I play an antibody right now. So this is the antibody, and then I stick a linker and a toxin to it. It's that small. So the chemistry is not that difficult, but to find out where the linker and the toxin sticks to the antibody, that's the real crux. And then, of course, you have a heterogeneous compound. So it's not a homogeneous compound. And this is why I think Chris and Lonza as well, we're looking into site-specific conjugations. So really to have a homogeneous compound where you only have 1 or 2 toxins sticking at the same position to the monoclonal antibody. But That's, I think we didn't touch that, but it's very, very important to say that the first generation of ADCs, and as you know, there's 2 ADCs on the market. It's Kadcyla, which has a blockbuster status, and there's a more niche product at Citrus from Seattle Genetics, which are on the market. But those are the old generation of ADCs. They are heterogeneous mixtures. However, until now, nobody could show that the site-specific conjugates are have more efficacy in the clinics, and that's we have to wait for. But that's where everybody's very nervous on. And I think, so let's see how the new generation of the ADCs will do. I sometimes hear people saying ADCs are dead. Clearly not. The pipeline is growing dramatically with these new technologies. But we also see a lot of new peptide conjugates, and we see compounds like combo therapies, you know, which are really, really exciting therapies. Again, you stimulate the immune system, and that's the beauty of it.
Yeah, that's a good point. I mean, we've been using the ADC term just because that has been the original technology, but certainly there's drug conjugates of other forms than the antibody that can be as effective or more effective. But I think also that kind of takes the conversation to the further on to the technology. So, you know, ADCs, again, you know, innovators often have the antibody, the target. They're going after the indication. They license or partner with the technology of the toxin and the payload. And so we talk about how can companies partner with technologies, and whether it's sort of part of the drug or part of the manufacturing process. So we talk about, okay, do we need different types of bioreactors to do these cell therapies? So moving from a 2D hyperstack, you know, plastic model in a hood versus a stainless steel system, and where do those technologies move? One of the interesting stories, Mark and I worked together at Millennium, and for those who may not know, the Accelerex platform originated out of that organization, and grew up, and now obviously it's a well-known and well-utilized single-use bioreactor platform. But it actually got its origins with an unmet need, and a product company initially funded it. There are other examples. So the Wave bioreactor came out of Shearing Plow. The HiClone system came out of Baxter. So again, these companies had unmet needs in the marketplace, actually were able to do that in-house, realize that they don't want to be technology companies in that sense from an equipment manufacturer, and spun those out. But I'd like to hear, where do you see that going? Are CMOs willing to make that investment, or Mark, is it really going to have to come from the product side to kind of figure out how do I build the better mousetrap and then have somebody ultimately take it over?
That's a good question. I mean, so it's a bit of both, right? I mean, I think what we, for as much as we are aspiring to be a product company, right, there's been a lot of investment in just technologies, right? So we know in order to improve our product yields, in order to improve some of the analytics and what we want to try to characterize, we're internalizing that, trying to partner with folks, academics, companies that have technologies, because part of it is, I guess we're looking at this as if this were biologics and more traditional antibodies, in the late 80s, right? I mean, again, I remember way back when we were trying to get to a gram per liter, all right, and that was a big deal, all right. And as far as the analytical technologies, I mean, again, the ability to characterize today is so profoundly different than it was when we had in at that time. And so our view is, okay, well, it's going to catch up. You're either going to be part of it or you're going to be left behind. There's going to be an element where the regulatory, as soon as it's available, regulatory expectations will go there, and so you have to be there. And so I think we're doing a bit of both right now. So I think the impetus is starting from internal. I think as and Andreas and Chris can add, I mean, as their companies start seeing that across platforms and across companies, then I think there's much more of an impetus for them to drive that as well.
No, absolutely. I mean, innovation is in principle comes by the innovators and their ideas, and we have to adopt our processes to their needs. But in these therapies, it's very important that it's not just the product, as we talked about, it's the process with all its supplements, the media, the growth factors, the bioreactor or the 2D reactor. There's a lot of things which come together. So in principle, every process is individualized and optimized together with the client. And again, it's very important that you see the synergistic effects of a CMO who can, of course, who can leverage the experience he has gained over the years by working with these clients together. And this is very, very often I get asked, wherefore do I need a CMO? And I think this is a point which is often neglected. A CMO works with so many different technologies from so many different innovators and partners. And at the end of the day, you can use the best techniques to make your clients successful. And I think this is very important, and even more important in those technologies where the process plays such an important role. And I would totally agree with Mark. Right now in cell and viral therapy, we are where mammalian business was 15, 20 years ago. We will see huge improvements in the productivity.
I think it's all well said. We are investing in technology to convert from 2D to 3D, as Lonza does.
So I think a combination of understanding technologies, how to apply them to the product and the process, understanding the potential variability as you might change the process. How does that all play into, once you've established proof of concept or been given fast track status, for example, your knowledge of the product and your process is always evolving. Even after it's commercial, you're still learning. So what are some of the things that you struggle with now? Because a lot of these emerging therapies are really being granted these fast track statuses because they are highly unmet medical needs, very innovative, and are showing to be very efficacious in early small trials, at least. So Mark, what keeps you up at night when you think about when you get fast track status and your CMC is 2 years behind?
So it's interesting. So we're in a number of programs like that, right? So it was interesting because even early on, so for example, within the EMA, there's an adaptive pathway process, right? So you can go and accelerate, but historically, whenever these have come up in regulatory settings, the acceleration is often in the context of clinical. And so it's clinical data. There'll be, a little bit more, we can get creative on how we do the designs, the numbers, but oftentimes what I've seen historically, again, is just from a CMC perspective, from a quality perspective, they're still holding the line, right? And so at least what we've seen, again, and this is in gene therapy, is that we've been very upfront about that, right? Saying, so if you want to get creative and get adaptive and get accelerated, you're going to have to get creative and adaptive on some of the quality and CMC aspects as well, right? So it doesn't mean cutting any corners. It just means when that certain information will become available, what is necessary versus what is nice to have from a more traditional product. And again, at least what we've seen is that because they're seeing the potential for the reward for patients being kind of a step function, they're being much more creative in working with us, right? So I think it does still give us nightmares because everything is accelerated, but I think they're being flexible. I do think what they're, I think this gets to the earlier question around this product versus process. The agencies are having to manage the fact that we're going to change these products throughout the life cycle. They're having to do it. Even though they're complex and maybe more complex than what we've seen in the past, they're going to have to deal with it because they see the advantages of literally what we're learning in the clinic over a small study of 15 patients. We can adapt and possibly get an even better product. And so the agencies see that, and so again, there is this, this collaboration that does happen. So at least we're seeing they're being open-minded to it.
There's not a lot more to add. I think I would totally agree. It's a dilemma. We have a lot of processes which are in late clinical phase with an older manufacturing technology. So probably the launch will happen with those technologies. But at the same time, we're talking to the agencies to switch over to a new modern technology. For example, moving from 2D to 3D processes. But this is something, right now, of course, many of the validated processes using 2D processes, and so there will be a scale-up issue in the future if the patient population rises, and so the authorities have to be open to switch the technology, and we are in a lot of discussions, and we see a lot of openness, especially, of course, in EMEA and, of course, in the Japanese market.
It's all well said. In the end, it's all well said. In the end, it's still the sponsor, the innovator, need to decide whether to take risks now or take risks later on. It's a balance of that.
Right, exactly. And I think, the whole QBD, investing in that early on certainly helps you accelerate the CMC process if you are given this opportunity to potentially file sooner and at the risk of not having exactly a complete package that you otherwise would have if you had the full development cycle time. So I think you're right. The dialogue is open with the agencies. My understanding is they are very receptive to this because they do want to get these products to the market, but they also want to get them to the products in a safe manner. So I think looking at it from that perspective is really going to be important. So I guess just one or two more questions, and then we can open up to a Q&A. But one of the other concerns really is around forecasting. So as we know, these are emerging technologies. They're still very early in the clinic. There's usually a lot of excitement with some early phase data, and then some not so exciting news as they go into larger trials and maybe have some clinical failure. So, that certainly affects the CMO business model because you're planning for a commercial launch, you're ramping up all these resources, and then the product fails. So let's talk a little bit about how you deal with that, the forecasting. So how big is, the ranges of probability are enormous, and thinking about that early on, how do you build your network? Where do you build your capacity, both physically in one location or in multiple locations? So again, we can start with Mark and work our way.
Sure. So it's a good question. I mean, especially, again, I'll speak for Bluebird, but as far as the way that the portfolio has been set up, it's relatively small early and then progressively gets to larger indications, right? And so again, for us, again, we made the choice to be virtual at this point to date, and that was because our lead indications were our smallest indications, so ultra rare, kind of smaller indications. We do see, the value at some point of essentially building capacity, but that capacity is going to be driven by something of volume, or at least a correlation of a few assets that have volume, that provide that sort of volume. But again, I mean, that's where, again, I think for us, the last several years, we've seen, we term the, we've grown to the right, which is kind of towards commercial and towards building that, bringing these products to market. We've had to grow to the left, right? We've had to grow from a platform perspective because, again, if you think of a sustainable model, the sustainable model has to have more products, right? It is enough capacity or enough volume of products to sustain a certain capacity and provide value. And so again, I think, for us, it's been trying to grow both to try to achieve that, right?
There's one beauty with the cell and viral therapy in comparison, for example, to the monoclonal antibody manufacturing. You don't need as big and piped facilities. You have more or less a clean room, which you can build up and which you can, thanks to single-use equipment, use for many different manufacturing processes. So I think it is important that as a CMO, you enable a shell and a room for potential candidates, but the risk you run is by far not as high from a capital investment as it is for a mammalian manufacturing side, where you have to spend $300 to $400 million. So I think you have to take a certain risk as a CMO, and I think Wuxi and Lonza are doing this. But the risk, if there is a clinical failure with these products, is not as high as it is, for example, for the mammalian manufacturing.
Totally agree with what Andrea said. Also, I think I mentioned this earlier. For CMO, because we're able to manage multiple programs, the risk is in a way is less because average out. And also, right now, the industry, based on the current industry outlook, still there are many more programs that come into the clinic. So even if the leading phase 3 struggles, the other programs actually need to come in very quickly. I think right now the industry average hold time is about 12 months. Right? You have to wait 12 months to go into the clinic. Yeah. So short term, there's not an issue, but long term, manufacturing, managing capacity is always a concern, even for not only for CMOs, but also for innovative companies.
We see that in a lot of our research as well, that capacity, for different types of products is potentially could be constrained depending on a few of the indication types that if they hit, certainly at the bioreactor type capacity for sure. So one last question before we do Q&A. Just interested, we talk a lot about capacity, technology, and things like that. What other bottlenecks do you see that are between you and advancing these therapies through the clinic? What about access to talent and things like that? Access to capital for, expanding your pipeline and other regulatory things, but maybe, talent in particular, both from a sponsor and a CMO. These are new technologies, and there's not maybe as deep a bench out there in the industry, and how are you dealing with that to make sure you have the best trained people to work on these products. You're potentially competing with each other for talent as well.
Yeah. So again, I think it's pretty common, right? I mean, so again, if you look back 5 years, 6 years with Bluebird, we made a choice where the talent was being acquired at that point from academia, right? Because these were technologies that were growing up in academia that at that point for us, it was not about getting to GMP and getting kind of all the bells and whistles as fully. It was more around getting the technology kind of secured. I think as we've grown as a company, what you do is you've started adding a lot of pieces towards the end game, which is commercial, right? So that is something which comes with bigger companies and experience. They are learning the technology and the nuances, but there's, again, you try to find the analogies. It's a rare disease space. It is small unmet medical needs. So you're trying to find some analogies that can play and be leveraged. But again, I mean, and that's where, again, I think the last several years, AAV companies have taken off, LVV companies have taken off. You've seen gene editing companies, and so if you look just in the kind of the atmosphere or the environment within Boston, they're all fighting for the same people, right? And so again, that's where again, if you've created an environment, you try to retain them. But again, that's been the difficulty probably the last 5 years. Is just how do you grow? How do you sustain that growth with the technology just changing so fast?
I would totally agree with you. There is a war for talents in this business right now. But having said this,
Can't hear. Yeah, I don't know.
Is it on?
Yeah, I don't know what happened.
Thanks, Mark. Hey, this one is so better. Did you understand me before? No. Great. Okay. Yeah, I would totally agree with Mark. There's a war for talents right now, especially in these areas. But it's also very, very important that you train the people very properly. And when you think about, I just give you an example about CAR T manufacturing. You take the blood from the patients, you genetically modify the blood. At the same time, the patient is treated with chemotherapy while your people are processing these modified cells and growing them, and then you have to re-inject them into the patient. There's a lot of stress on the labor, and you need to train these people extremely well because it's one shot. And you have to deliver the right quality in the right time. And this is extremely important, and that's why I believe it's all about training. In terms of other bottlenecks, of course, we have a scale-up bottleneck, but I wouldn't say there's no problem I can see which cannot be solved in the next couple of years. There's nothing, and again, many of these treatments, they treat unmet medical needs, and as long as they do, there will be always a solution.
Hello. I think maybe it's this one. This universal one now. I think the bottleneck, besides talent. So I agree with Mark and Andreas. Fighting for talent or competing for talent has always been the case. I think hiring the right people and then train the new people, get them up to speed very quickly. That's absolutely important. Besides the talent, actually capacity. I think right now there's still very limited capacity on the cell therapy and gene therapy side. So either the established players invest more, or the new players coming into come into this industry. It's very important to support the future growth of the industry.
Okay. All right, with that, I'd like to open up for Q&A. You can't hear me either? Maybe that's just hold it up closer. So there's a microphone there. Please use it, obviously, because it's hard to compete with the convention floor. So please maybe just state your name and your affiliation and your question.
There we go. Hi, guys. Thanks so much for this great panel. I just wanted to ask a really simple question based on what Andreas had mentioned. How bullish are you in the looking forward towards gene and cell therapy? You mentioned that it's taken many years to come up with a homogeneous product for ADCs, and cells, if anything, are going to be even more heterogeneous as we modify them and do things, and even select for the right population to begin with. Do you think that this is going to take, I mean, it's clear that the impact will be there, but do you think it's going to take, half a century to get there?
Yeah. So I mean, it's a good question. So I think what's different, right? I mean, so what's maybe different is you're looking at indications across the board, especially you're talking about gene therapy or chimeric antigen receptors. I mean, these are profound step-ups of risk-reward, right? And so will they be perfect products the first time out? No, but will they be products? Absolutely. Right, so I think this data set's out there. I mean that these drugs are making a difference. So I think they are products. I think that you're seeing over the last 3 years and more companies join because it is that second wave, right, that you typically see, which is now that you've got some products. I mean, you've seen also big changes in the risk profile. There's been a lot more understood about insertional risks and things like that over the last 10 years. So I think there's a couple of big hurdles that are starting to get addressed, and now you're seeing what hasn't been seen, which is the profound effect to patients, which we started seeing in the last 5 years. And so that's why, again, I think we feel strongly it's different this time around, right?
There's not a lot more to add. I mean, you saw the first videos about curative approaches. I think you get goosebumps when you see these videos, and I mean, that already excites me every day when I come to work. There is a new kind of products coming onto the market which have curative effects. You use your own immune system to fight the diseases. I mean, this is exciting. This has never happened. And the side effects, when you look into videos of patients who get treated with ADCs, I mean, the side effects, they can have a normal life. And this is remarkable. So I think it hopefully will not take as long, but we need a few next shots, especially also on ADCs. We will see those will come, but hopefully not in a decade.
If you just say your name and your company.
Hi for me. My name is Maria. I'm from a startup in Taiwan called ImmuneWork. We are working on a new ADC platform that may solve some homogeneity problems. So according to your expertise, how soon do you think we should bring in some processing expert to help us, during the development process so we can, get it fast on to the clinical trials?
I'm fair. I leave the to you first.
I think as long as you have some ideas, you really need to look at the processing, because as Andreas mentioned, ADC processing is actually key. But as early as possible.
This time, nothing to add. So.
Yeah, I think assembling the right team early is always important because it's getting it understood, understanding your process as well as you can early on really helps minimize risk down the road. So when you do make process changes, you understand what you are changing and you're intentionally doing it, not unintentionally doing it, and have control over that, and so that you don't have to redo or invalidate some of your clinical results or even your non-clinical results. That can be very timely and costly just to get into the clinic as well.
Hi, my name is Manal. I'm a PhD student from the RWTH Aachen University. I
studied in Aachen.
Oh, so. Yeah? So, yeah, I did my PhD in recombinant therapeutic proteins, and a constant issue is always like these failings of these therapeutic biologicals in clinical trials. And I was wondering, like, how is it different from chemicals that are going through clinical trials? Is the ratio of failing to and successful different for the biologicals rather than the chemicals?
Yeah, I mean, there's a lot of literature on this, right? I mean, so with small molecules, there's an expectation, right? I mean, I forget what the numbers are today, but it's whatever 10% will make it through, and a lot of it's because you'll have preclinical failure, you'll have your safety failures. Biologics, I think the numbers went up and the numbers staggered where there's different failure points. I think even more recently I've seen numbers where, in rare diseases, the numbers are higher than in more traditional chronic indications, just because, again, the risk reward and some latitude that's allowed there. So I think at least some of these novel ones, it's still to be played out. I think there's not a lot of history yet to say what those true numbers are. I think there's some areas where we benefit, where it's again a monogenetic disease. If you're able to fix that one, specific mutation, right, you should have an effect, right? So again, but now will that translate to some larger failure rate downstream? That's to be seen. So again, so I think that they're well played out, and I think that they all vary around the same. I think what they vary is where the failures occur.
No, you make a great point, Mark. It's really hard to predict. And right now, we would love to have more reliable data. I mean, in 1990, it was like 1 out of 4 products made it from phase 1 onto commercial. Right now, it's 1 out of 8 in a total average biologics and small molecules. But when I look into cell therapy, for example, there is no failure rate from the phase 1 into phase 2. You directly move into efficacy because, I mean, these things are your own stem cells often, so they are safe from a safety profile. But overall, when you look into... now I lost what I want to say, but maybe I remember it later on.
I don't have anything to add.
I think that the issue with the biologics, they tend not to, some proteins, for example, tend not to fail as early because they are safer in general, and certainly some of the cell therapies, which means you spend more money longer into the development cycle too. There's a terminology, kill quickly, kill early, in the small molecule side, because you want to know if it's going to fail quickly because you don't want to invest a lot of money. Unfortunately, that doesn't always happen in the biologics. You end up taking it very far into development, into large pivotal trials, and then spend a lot of money and then get the disappointing news. So it's tough. It is the risk and reward. How much are you willing to spend for that, to prove the reward?
I remembered what I want to say. Until 2010, there was a huge decline in new drug and BLA approvals. And since 2010, since the focus has shifted a bit more towards more targeted, personalized medicines, we see a huge uptake in the new drug approvals and BLA approvals. I think last year was a record year. It's maybe supposed to be that this year is another record year. But that also has to do with the changing focus of the pharmaceutical companies. It's not only the focus on blockbusters anymore. It's more the focus on really targeted, personalized medicine.
Okay. No? Well, if not, then I will thank you very much for your attention. And maybe if the panelists can stay for a minute or two, if those who are too shy to ask questions want to come up and do it one-on-one, I'm sure that they'd stick around for a minute or two. Okay? Thanks very much.
Thanks, Patty.